Hello, everyone. Welcome to today's lecture on cell biology, specifically focusing on tissue development and morphogenesis. In this lecture, we will delve into the intricate processes that govern the formation and organization of tissues in multicellular organisms. We will explore various topics, including embryonic stem cells, cell signaling pathways, induction and competence, epigenetic regulation, cell adhesion and migration, organogenesis, pattern formation, morphogen gradients, the role of extracellular matrix, stem cell niches, and the influences of genetics and the environment in tissue development. Let's begin with our first topic. Tissue development is a complex and dynamic process that involves the coordinated actions of cells, signaling molecules, and genetic programs. During embryogenesis, a single fertilized egg undergoes a series of cellular events to give rise to various cell types that eventually organize into distinct tissues and organs. This process, known as morphogenesis, is tightly regulated and relies on the precise control of cell fate determination, proliferation, migration, and pattern formation. Embryonic stem cells are pluripotent cells derived from the inner cell mass of a developing embryo. They have the remarkable ability to differentiate into all cell types of the body, making them valuable tools for studying tissue development. Through controlled manipulation of signaling pathways and transcription factors, embryonic stem cells can be directed to differentiate into specific cell lineages, mimicking the early events of tissue development. Cell signaling pathways play a crucial role in tissue development by transmitting information between cells and coordinating their behaviors. Signaling molecules, such as growth factors and hormones, bind to receptors on the cell surface, initiating a cascade of events that regulate gene expression, cell proliferation, and differentiation. Understanding these signaling pathways is vital for unraveling the mechanisms underlying tissue development. Induction refers to the ability of one group of cells to influence the fate of neighboring cells. Through various signaling mechanisms, including secreted molecules and cell-cell contact, inducers can stimulate the expression of specific genes in responsive target cells, altering their developmental trajectory. Competence, on the other hand, refers to the state of a cell or tissue to respond to inductive signals. The intricate interplay between inducers and competent cells is essential for proper tissue development. Epigenetic regulation refers to heritable changes in gene expression that do not involve alterations in the DNA sequence. During tissue development, epigenetic modifications, such as DNA methylation and histone modifications, play a critical role in controlling gene expression patterns. These modifications can confer stability to cell fate decisions and ensure the proper differentiation and function of tissues. Cell adhesion and migration are fundamental processes in tissue development. Adhesive proteins, such as cadherins and integrins, mediate cell-cell and cell matrix interactions, enabling cells to form cohesive tissues and navigate through complex environments. Cell migration is essential for tissue remodeling, organ formation, and the establishment of proper tissue architecture. Organogenesis refers to the formation and differentiation of organs from embryonic tissues. This process involves intricate interactions between multiple cell types and requires precise spatiotemporal regulation of cellular behaviors. Key events in organogenesis include tissue patterning, cell differentiation, and the establishment of functional tissue structures and connections. Pattern formation is the process by which cells organize themselves into spatially distinct regions, giving rise to the characteristic shapes and structures found in tissues and organs. This process involves the establishment of morphogen gradients, which provide positional information to cells and guide their fate and behavior. Mathematical models and genetic studies have shed light on the mechanisms underlying pattern formation during tissue development. Morphogens are signaling molecules that form concentration gradients within developing tissues. These gradients provide spatial cues that guide cell fate determination and cell behaviors. Through processes such as diffusion and receptor-mediated signaling, cells can interpret the morphogen gradients and respond accordingly, resulting in the establishment of tissue patterns and structures. The extracellular matrix, ECM, is a complex network of proteins and carbohydrates that provides physical support and biochemical signaling cues to developing tissues. The ECM not only helps to maintain tissue integrity but also plays a crucial role in modulating cell behavior, including proliferation, migration, and differentiation. Alterations in ECM composition can have profound effects on tissue development and homeostasis. Stem cell niches are specialized microenvironments that support the maintenance and self-renewal of stem cells throughout development. These niches provide essential cues, including growth factors and physical interactions, that regulate stem cell behavior and fate decisions. Understanding the niche dynamics is crucial for harnessing the regenerative potential of stem cells in tissue repair and disease treatment. Tissue development is influenced by a complex interplay between genetic and environmental factors. Genetic programs dictate the intrinsic properties of cells, while environmental cues, such as nutrient availability and tissue mechanics, shape cell behaviors and tissue morphogenesis. Understanding the interaction between genetics and the environment is essential for comprehending the variability observed in tissue development and disease susceptibility. In conclusion, tissue development and morphogenesis are fascinating processes orchestrated by a myriad of cellular and molecular events. From the differentiation of embryonic stem cells to the establishment of tissue patterns and the influence of genetic and environmental factors, each step contributes to the intricate fabric of multicellular life. By studying these processes, we gain valuable insights into human development, tissue regeneration, and disease mechanisms, paving the way for future discoveries and medical advancements. Thank you for your attention, and I look forward to our next lecture.